Hi all, here is the chapter 22 overview video for you. In the Galapagos Islands, Darwin discovered plants and animals found nowhere else on Earth. So he kind of discovered these different species. So speciation is a process by which one species splits into two or more species. And this explains the features shared between organisms due to inheritance from the most recent common ancestor. Speciation kind of forms a conceptual bridge between microevolution and macroevolution. So remember, microevolution consists of changes in allele frequency in a population over time. Macroevolution refers to the broad pattern of evolutionary change above the species level. So this is talking about new species being formed over millions of years. The biological species concept states that a species is a group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring. They do not breed successfully with other populations. So what is a species? A species is a group of individuals or members that interbreed and produce viable offspring, and they do not breed with other populations and create viable offspring. Gene flow between populations holds the populations together genetically. So there's different ways that species are maintained, and we talk about that through reproductive isolation. So that's the existence of biological barriers that impede two species from producing viable, fertile offspring. Hybrids are the offspring of crosses between different species. So a hybrid is like a mule, a cross between a horse and a donkey. You've heard of ligers, a cross between a tiger and a lion. So those are very rare, but they do occur sometimes. Reproductive isolation can be classified by whether barriers act before or after fertilization. Pre-zygotic barriers block fertilization from occurring. So pre is before. So they block fertilization by impeding different species from attempting to mate, preventing the successful completion of mating, or hindering fertilization if mating is successful. So there can be, a form of pre-zygotic isolation is habitat isolation. Two species encounter each other rarely, or if at all, because they occupy different habitats. So they are geographically in different places. Temporal isolation, species that breed at different times of the day, different seasons, or different years can't mix their gametes. So this is like space, this is like time. And then the third one is behavior. So courtship rituals and other behaviors unique to a species are effective barriers. So if one bird has a particular song, another female bird of a different population is going to be like, ew, yeah, no. So they are not going to interbreed. So here are some of these pre-zygotic barriers that we just talked about: habitat isolation, temporal isolation, and behavioral isolation. Mechanical isolation is another pre-zygotic isolation method. And this is when there's morphological differences that prevent successful mating. So something with the anatomy prevents two species from mating. Gametic isolation is that sperm of one species may not be able to fertilize eggs of another species. And I have a video to show you guys, but it will do that during one of our Zoom sessions. All right, so those are the last two pre-zygotic barriers. Post-zygotic barriers prevent the hybrid zygote from developing into a viable, fertile adult by reducing hybrid viability, reducing their fertility, or it's called hybrid breakdown. So in reduced hybrid viability, genes of the different parent species may interact and impair the development or survival of the hybrid. Reduced hybrid fertility, even if the hybrids are healthy, that's what it means by vigorous, they may be sterile. So this is true of most hybrids. So mules are sterile, ligers are sterile, so they cannot mate and produce offspring. And then hybrid breakdown is that some first generation are fertile, but then when they mate with another species or with either parent species, offspring are feeble or sterile. So it's just saying that their offspring, even if they can create offspring, if they're not sterile, that their offspring then are probably going to be sterile or not really viable, not really strong. So those are the 3 post-zygotic barriers. There are limitations to the biological species concept, so it can't be applied to fossils or asexual organisms, including all prokaryotes. And it does emphasize the absence of gene flow, but that's our best way to define what a species is. All right. Speciation can occur in 2 ways. So we just talked about ways that species are maintained. And now we're going to talk about how that speciation can occur. So there is allopatric speciation and sympatric speciation. So here's this pond of these fish, and allopatric speciation forms a new species while geographically isolated. So if this little berm goes all the way up, we have this separation, and then speciation will occur between these 2 species. Sympatric speciation occurs without geographic separation. So for some reason, those 2 populations just decide not to interbreed, and then they become different enough that they form 2 species. In allopatric speciation, gene flow is interrupted when a population is divided into geographically isolated subpopulations. All right, so, and it depends, geographic barrier depends on the ability of a population to disperse. So a canyon may create a barrier for small rodents, but birds could travel over, coyotes may be able to navigate that. So it just depends. And then separate populations may evolve independently through mutation, natural selection, and genetic drift. And then reproductive isolation may arise as a result of genetic divergence. So our example here is mosquito fish in the Bahamas comprise several isolated populations in different ponds. So you can see just the shape of the fish is different. So under high predation, the body shape enables rapid bursts of speed is favored, where then under low predation, the body shape that favors long, steady swimming. So they developed 2 kind of separate populations. And evidence of allopatric speciation is 15 pairs of sister species of snapping shrimp are separated by Panama. And these species originated from 9 million to 3 million years ago when Panama formed and separated Atlantic and Pacific. So you can see here's Panama and then all these different species of snapping shrimp. So regions with many geographic barriers typically have more species than do regions with fewer barriers. Reproductive isolation between populations generally increases as the geographic distance between them increases. So there's not as much chance for interbreeding. In sympatric speciation, speciation takes place in populations that live in the same geographic area. So it occurs when gene flow is reduced between groups. And the main factors for sympatric speciation are polyploidy, habitat differentiation, and then sexual selection. So polyploidy is the presence of extra sets of chromosomes during accidents during cell division. It's way more common in plants. An autopolyploid is an individual with more than 2 chromosome sets that's derived from one species. And then the offspring of matings between autopolyploids and diploids have reduced fertility. So let me just show you what this looks like. So a cell division error occurs and we end up with a tetraploid cell. So instead of 2n equaling 6, now we have a 4n cell, which equals 12. These gametes then are going to have more, they're going to be diploid. So they're going to produce 2n gametes, and then we're going to wind up with this new species of 4N. Again, typically this only happens in plants. So allopolyploids is a species with multiple sets of chromosomes derived from different species, and they can't interbreed with any parent species. So crops like oat, cotton, potatoes, tobacco, and wheat are all polyploids. So you can see if we have species A, this is like our polyploid here, or could be an autopolyploid. Anyway, some sort of polyploid individual breeds with a different species that has 4. So, okay, I said that backwards. Here's our regular species, right? Here we have a mitotic error. So where we have unreduced gametes, we combine these, and then we have a normal gamete, and we have a gamete that has way too many chromosomes. This then might produce a new species. So all of this is due to chromosome error, but because there's more chromosomes, that creates a new species. And there's also habitat differentiation. So sympatric speciation can result from the appearance of new ecological niches. So a maggot fly, for example, can live on hawthorn trees or apple trees. And the flies that then live on the apple trees can create a new species. Sexual selection can drive sympatric speciation as well. An example is sexual selection for mates of different colors has likely contributed to speciation in fish in Lake Victoria. So you can see we have normal light, and then orange light creates these 2 different looking species. In allopatric speciation, geographic isolation restricts gene flow, which then can lead to new species being formed. By reproductive isolation can arise by natural selection, genetic drift, or sexual selection in those isolated populations. Even if contact is restored between the populations, interbreeding is prevented then by those reproductive barriers that we talked about at the beginning. In sympatric speciation, a reproductive barrier isolates a subset of a population without geographic separation, and that can result from polyploidy, natural selection, or sexual selection. All right, hybrid zone is a region in which members of different species mate and produce hybrids. Hybrids are the result of mating between species with incomplete reproductive barriers. So it can occur in a single band where adjacent species meet. So you can see right here is a hybrid zone between fire-bellied toad and yellow-bellied toads. There's the difference you can see between the 2 toads. Hybrids often have reduced fitness compared with parent species, and the distribution of hybrid zones can be more complex if parent species are found in patches within the same region. So within a hybrid zone, there's 3 possible outcomes. There could be reinforcement, fusion, or stability. Reinforcement occurs when hybrids are less fit than the parent species, so then the parent species are reinforced so that hybrids are not selected for. Fusion is where the hybrid is a mix and the parent species are kind of mixed into a single species. That may occur if hybrids are as fit as parents, allowing substantial gene flow. So it would kind of be like a cross between 2 species or a fusion. So you can see here's like hybrid offspring. And then stability may be achieved if extensive gene flow from outside the hybrid zone can overwhelm selection for increased reproductive isolation inside the hybrid zone. So hybrids are continuing to be produced, but they're not increasing or decreasing. It's just kind of a maintenance. So stability is just kind of where they're maintained. So here we have a population where gene flow is occurring, and then an isolated population diverges, and then we have 2 populations that develop. two separate species. And then between these two is the hybrid zone. And again, the possible outcomes are reinforcement, where the two parent populations are maintained, fusion, where those two parent populations actually become one population, and stability, where we're just kind of maintaining the parent populations, and then a few hybrids are produced. All right, so many questions remain concerning how long it takes for a new species to form or how many genes need to differ between species. Broad patterns in speciation can be studied using the fossil record, morphological data, or molecular data. The fossil record includes examples of species that appear suddenly, persist, essentially unchanged for some time, and then apparently disappear. So these periods of apparent stasis, or kind of like nothing's happening, punctuated by a sudden change, are called punctuated equilibria. So there's this model, punctuated equilibria or punctuated evolution, and then there's gradual evolution. So you can see in the punctuated model, there's a change that occurs very rapidly that's maintained, and then gradual model is we have these intermediate kind of species before we get to present day. So two different models of evolution. The punctuated pattern in the fossil record and evidence from lab studies suggests that speciation can be rapid. So sometimes that does happen. So the interval between speciation events can range from 4,000 years, which is really fast, to 40 million years. So the average is about 6.5 million years for a new species to develop. And there's our example with the sunflowers. All right, hope that helps. Bye, guys.